Hi guys, a very warm welcome to all of you in our Gurmantra Academy. I'm your educator Saranya, and as you know, we are seeing the immunology lectures. So today we are going to see some more topics relevant to immunology. Okay, so today's topic is antigen processing and presentation, that is called as APC. Okay, antigen processing and presentation. And how does it happen? There are two ways for this purpose. Okay, so usually if the antigen is endogenous in nature, okay, then that will undergo proteasomal degradation and the peptides will be about 8 to 10 amino acids, and that will be represented by your MHC class 1, which will be identified by TC cell receptor, that is CD8. So this is for endogenous antigen. But if the antigen is exogenous in nature, that will bind to the receptor, it will be taken inside, and processing will happen. Processing is where this particular bacteria will be broken down into smaller pieces, which will be of 13 to 18 amino acids. Okay, 13 to 18 amino acids. And how is this degradation taking place? By endolysosome pathway. What does that mean? आपका जो बैक्टीरिया सेल के अंदर जाता है, देन इट विल गो इनसाइड एंडोसोम एंड फ्यूज विद लाइसोसोम. Because lysosome has so many hydrolytic enzymes and the pH is very low, it will degrade the molecules up to 13 to 18 amino acids. So if it is 13 to 18 amino acids, that will be represented by your MHC class 2, which will be recognized by T helper cell, which has the receptor CD4. Okay, so this is the process of antigen processing and presentation. How is it processed? Two ways are there. Either it is processed by using endolysosomal pathway. Okay, so this is your part 1. Second one is proteasomal degradation. So these two are the pathways through which your antigen will be processed, and based on what is the number of amino acids a particular antigen represents, it can be either your MHC1 or MHC2. Okay. Okay, so when we talk about MHC1, MHC1 is always endogenous in nature. You have to remember this. Okay, MHC means endogenous in nature. With MHC1 will be presented by all the nucleated cells which has nucleus. So like for example, endogenous antigens are something proteins which is present inside your body itself, not a foreign particle. Like for example, you have a misfolded protein which has been not folded properly, and it can be causing diseases like prions. Okay, it can be like prions. So misfolded protein that will act like an endogenous antigen. Now this will go inside the proteasome and it gets degraded to 8 to 10 amino acid antigenic peptides. Okay. After this will go to the endoplasmic reticulum. In endoplasmic reticulum, MHC1 will be present. Okay, MHC1 will be present. So MHC1 assembly with the antigen, that is your 8 to 10 amino acid antigen plus your MHC1 molecule will be assembled into a single unit. Okay, and it will be packed inside a vesicle. Can you see here? It is packed inside a vesicle. Now this will be exocytosed from the ER. It will take exit from ER and go to Golgi. From Golgi again, it will take the form of a vesicle, and now it will go to your plasma membrane. Okay, vesicle will be removed. In Golgi, vesicle will be removed. Your MHC1 along with the antigen will go to the plasma membrane. Okay, in the plasma membrane, it will be represented. Like for example, this is your cell and this is the plasma membrane, means it will be represented like this. Okay, let's say this is your MHC1 and this is your antigen. It will be represented like this. So what is the T cell that is responding to your MHC1? That is your TC cell. Now let's say this is a TC cell. It will come, which will have a CD4 receptor, CD8 receptor, and that will recognize this antigen and receive the signal. It will receive the signal that there is an antigen. So antigen processing as well as presentation is done using MHC1. Okay, can you see here? Can you see in this picture? Proteasomal degradation. For what? For MHC1. MHC1 proteasomal degradation. Just these are some of the key points you have to remember. Okay, because all of these are related. Like a revision box you can use. Okay, MHC1 proteasomal degradation. Okay, MHC1 proteasomal degradation. Then you have endogenous antigen, TC cell, CD8 plus. So these are some of the terms you should actually know. These are some of the terms you should know, which is like a revision box, like keywords for your this particular MHC1 representing pathway. Okay. Next is MHC2. MHC2 directly means your, okay, I want to add one more point to this, that is 8 to 10 amino acid. Okay, this is one more point I wanted to add. Next is your MHC2, which is exogenous pathway. It will follow exogenous antigens, which means the antigen that is coming from outside of your body. Here the antigen processing and presentation is for 13 to 18 amino acid antigens. Okay, antigen of 13 to 18 amino acid. So you have an antigen and an antigen presenting cell. First step is this particular antigen will be endocytosed, which means let's say if this is an antigen and this is a cell, okay, this will go inside this like this. Endocytosed, it will go inside and the vesicle will form and it will go inside. Okay, endocytosis of antigen via a vesicle. Okay, now it will fuse with another vesicle, which is nothing but your endosome. Okay, it will fuse with endosome. Endosome is one of the important organelle which will start your hydrolysis. Okay, so what happens? It fuses with endosome. pH in the endosome is very low, which means it's very acidic. Okay, now this endosome will fuse with your lysosome. Endosome pH is low. Lysosomes has hydrolytic enzymes. So what it will do? It will break the bacteria or the antigen into small peptides of 13 to 18 amino acids, which will be present into endolysosome complex. Okay. Now, along with this endolysosome, your MHC class 2 will come and fuse. Okay, MHC class 2 will come and fuse. Basically, this MHC class 2, where does it come from? That also you have to know. Suddenly, it will not appear from somewhere, right? So, MHC class 2 will be assembled in the endoplasmic reticulum, but the alpha 1 and beta 1 unit will be blocked. Okay, alpha 1, beta 1. You know the structure of MHC class 2, right? So, this is one structure. This is the other structure. This is plasma membrane. Alpha 1, alpha 2, beta 1, beta 2. So this is the structure of MHC2. Can you see in the ER, this the antigen binding location. This will be blocked. Okay, this will be blocked using a clip. It will be blocked using a clip. Okay, so this MHC2 will be inside a vesicle. It will be assembled, but it will be blocked. It will go to Golgi. Then it will react with your antigen, which is of 13 to 18 amino acid in length. When it meets this kind of an antigen, the clip will be removed. The clip will be removed, and now there is a complete complex of MHC class 2 along with the antigen will be formed. Now this will go to the plasma membrane. It will be exocytosed. So let's say this is your cell. Okay, now here you will have your MHC class 2. with an antigen. You will have another cell which is, let's say, TC cell, T helper cell, that will have a receptor. What receptor? CD4 receptor, that will recognize this particular antigen and it will get activated. So this is the process of antigen processing and presenting of the exogenous antigen. So let's do the revision box. Revision box. First thing, MHC2 will be used. Then it will be exogenous antigen. Okay, it will be exogenous antigen. It should be 13 to 18 amino acid in length. And how is the degradation taking place? It is using endolysosome complex. Endolysosome complex is the thing. Okay, endolysosome complex will be used, and here clip will be present on alpha 1 beta 1 subunit of your MHC2. Okay, so this you have to definitely remember. Okay, like here we saw for MHC1, this is for MHC2. Also, this will be recognized by your T helper cell which has CD4 receptors. This alone you have to know. Okay, these are the key points. So can you see here, there is a bacteria that is engulfed. Okay, this is a bacterial pathogen that is engulfed by phagocytosis and phagosome is formed. To this lysosome will fuse. Okay, to this lysosome will fuse to form endolysosome. And to MHC these molecules will be attached, which is nothing but 13 to 18 amino acids long, and they will be presented on the surface in the plasma membrane. Okay, that is all about your antigen processing and presentation. Now let's move on to the structure of antibody. So what is antibody? Antibody is basically a protein. Okay, that is a protein. It is also called as immunoglobulin. It is Y-shaped and it consists of four polypeptide chains. They are 2 heavy chains, 2 light chains. Okay, they have several functions. One is antigen binding and elimination of the antigen. Okay, elimination of the antigen. So each function is carried out by different parts. So antigen binding part is Fab region is for antigen binding. FC region is for the crystallizable. It's for crystallizable biological activity mediation के लिए यूज होता है. सो फैब रीजन जो होता है वो एंटीबॉडी के साथ, मतलब एंटीबॉडी का जो फैब रीजन होता है अपर पार्ट ऑफ द रीजन होता है जो कि एंटीजन के साथ बाइंड करता है. ओके, इट विल हैव वन कांस्टेंट रीजन, वन वेरिएबल डोमेन with heavy chain and light chain. Okay, so this region is called as a paratope. Paratope is a region on antibody that will bind to the antigen. Epitope is a region of antigen which will bind to the antibody. ये डिफरेंस आपको डेफिनेटली पता होना चाहिए. FC रिसेप्टर क्या करता है? ये कॉम्प्लीमेंट सिस्टम में यूज होता है. ओके Okay, so antibody is also called as immunoglobulin, which is Y-shaped protein that will be fighting against, that will basically detect the antigen and it will remove it from our system. Okay, so it will be produced by B cell, B lymphocyte. Okay, it will be produced by B lymphocyte which will be differentiated into plasma cells and memory cells. Plasma cells will produce antibodies, memory cells will be ready for the reinfection. Okay, so basically there are 4 peptides. Okay, 2 identical heavy chain, 2 identical light chain. Light chain is around 25 kilodalton and 25 kilodalton, and heavy chain is 50 kilodalton. Okay, basically 5 types of immunoglobulins are present. You have alpha, you have delta, you have epsilon, you have gamma, and you have mu. You can remember it as GADME or GAMED. Anything, any way you can remember. All you have to do is remember the facts. This way you can remember. These are the heavy chain types. Light chain type is also present. One is kappa, the other one is lambda. Okay, these are the light chains. And the antibody is having a variable region and a constant region. So the variable region is the region to which the antigen binds, and the constant region will be present downwards. Okay, can you see this is the structure of a antibody? So this is your variable region. Okay, this is your variable region, and this is your constant region. This C and all is there, right? These are constant region. Okay, so this is heavy chain. This blue color is heavy chain, purple color is your light chain. Okay, purple color is your light chain. So this is the Fab region. Okay, antigen binding region. One antibody can bind to two antigen. You have to remember this. Okay, this is the antigen binding site, hypervariable region to which the antigen will bind. Okay, so this is the FC region. This is the FC region which is involved in the complement system. All of these regions are interconnected with each other using disulfide bonds. Okay, using disulfide bonds. So this is another picture of your your antibody, and with different domains. Again, different picture, but here I am going to tell you some important point. So up to this will be your Fab region, and this is your FC region. Okay, this is your Fab region, and downwards is your FC region. Okay, up to this Fab, then downwards is FC. Okay, remember that. So this is your variable region and hypervariable region. Okay, this is your FC region. Now we are going to use certain protease enzymes to cleave the antibody, and we should know where it cleaves. If you are going to use papain, it will cleave here. It will cut here. So you will get one FC region and two Fab region. Okay. Next is your papain. Papain will cut here. Papain will cut here. Then you will get one FC region and one Fab region that is attached with each other. Okay, so these are some of the things you should know because that will be asked. So classes and types of antibody. There are several classes and types of antibody. We actually saw GADME, your gamed, gamma, alpha, delta, mu and epsilon. Okay, first we are going to see about IgG. IgG means immunoglobulin gamma. Its molecular weight is 150 kilodalton. Okay, so two identical gamma heavy chains which is 50 kilodalton, and then you will have light chain which can either be kappa or lambda, which will be 25 kilodalton. Okay, so IgG provides a long-term protection from infections. Okay, for example, अभी ये जो कोरोना वायरस हुआ है हमारी बॉडी में, जब कोरोना वायरस एंट्री मारता है तो IgG का लेवल्स बहुत ज़्यादा इंक्रीज हो जाता है, एंड द पर्सन इफ ही इज रिकवरिंग फ्रॉम कोरोना वायरस, उसकी बॉडी में IgG लेवल्स मेंटेन होती है ताकि फॉर अ लॉन्ग टर्म उनकी बॉडी में अगेन री इन्फेक्शन ना हो जाए. ओके, नेक्स्ट इज IgM. IgM is a pentamer. Okay, IgM is a pentamer. It has 5 units. Okay, it has 5 units, which means 10 antigen binding regions will be there. Okay, and these 5 units will be linked together with each other using J chain. Okay, IgM is also involved in ABO blood group classification. Next is IgA. IgA is also called a secretory antibody because it will be present in secretions like breast milk. Okay, it will be present in secretion, and this is a dimer. Okay, it can either be a monomer or a dimer. Okay, it will be present in secretions. Next is IgE. IgE is mainly involved in your hypersensitivity reactions and for the elimination of parasitic worms. IgD. IgD is also expressed on the B lymphocyte. B lymphocyte will basically have IgD and IgM on it. Okay, and that will play a very important role in antibody production. So these are the structures. You can screenshot this and use it as a revision box. IgG is abundant and it is very, very important. Next is IgM, which is pentamer. Okay, IgG can also cross the placenta. IgA is secretory. You can see it is a dimer joined by J chain. Here also J chain is present. Next you have IgD and IgE. Okay. So function of antibody, it will provide long-term protection because memory cells along with this will also be formed. Second, they will neutralize the antigen that is produced by either bacteria, virus, trigger the complement system and enhance the phagocytosis, right? And also it is present, the antibodies is present in your tissues like secretions. These secretions will eliminate the antigen very quickly, okay? So IgA is the first line defense which is present in mucosa of the intestine, nose and lungs. IgM will be determining the ABO blood group. Okay, this is also involved in phagocytosis. IgE along with the basophils and mast cells will involve in hypersensitivity reaction. Okay, this also helps in parasite elimination. IgD will be present on B cell surface and helps in antibody production. Okay, so this is a very good differentiation table between different antibodies and its characteristics. Okay, these are the first is the structure. Second, heavy chain. IgM means mu, IgG means gamma, IgA means alpha, IgE means epsilon, IgD means delta. Okay, number of antigen binding sites for IgM it is 10. Why? Because it is a pentamer. That is why. For monomer there are 2 antigen binding site, means for pentamer there will be 10 and for dimer there will be 4. Okay, important MCQ questions. Next, molecular weight you don't have to remember that much, but you need to know which is abundant. IgG is abundant, and after that it will come IgA, then IgM, then IgD and IgE. Okay, what can cross the placenta? Only IgG can cross the placenta. MCQ question. Okay, what will fix the complement? IgM and G both will fix the complement. Important MCQ question. This is MCQ, this is MCQ, this is MCQ, and the structure of course is MCQ. Next, binds to FC binds to phagocytes and mast cells, and that is your IgG and EE. So functions you already know. Okay, so this is another one which I got it for you. Okay, you can also screenshot this and use it for your revision purpose. Okay, so now we have few questions. Okay, antibodies are, what are they? They are glycoproteins. Okay, because they have that is carbohydrate moiety also. They are basically proteins, but they have carbohydrate moiety. That's why they are glycoproteins. Light chains and heavy chains are joined with each other by what? Do you know the structure? So this is the structure of basically antibody. This is heavy chain, this is light chain. How are they joined with each other? I showed you in the diagram. Do you remember this? How are they joined with each other? You see the diagram and tell me the answer. Can you see how are they joined with each other? Here you can see the heavy chain and the light chain are joined using disulfide bonds. Okay, disulfide bond is the correct answer for this question. Okay, so I hope you guys would have liked this class, and if your friends are also interested, you can ask them to join our lectures by using the code, referral code Saranya10. Okay, so this is our academy, Gurmantra Academy. You can visit the website. You can join our Telegram channel, Gurmantra Academy. Okay, and if you have any queries, you can always contact us. Thank you so much, guys. Have a nice day.